Hello students, today we are going to introduce DNA sequencing method. First introduce what is meant by DNA sequencing. The determination of nucleotide or base sequence of DNA we call as the DNA sequence. We have developed several different methods for the DNA sequencing like Maxim Gilbert sequencing, chemical sequencing, Sanger sequencing, dideoxynucleotide chain termination, automated DNA sequencing, pyrosequencing, Ruche 454, solid sequencing, and Illumina sequencing. So we can use any one of these methods for the sequencing of our DNA templates. So today we are introducing the pyrosequencing method. It is one of the methods used for the sequencing of DNA templates. This method is based on the detection of pyrophosphate, which get released during the DNA synthesis due to the addition of nucleotide whenever any nucleotide is get added during the DNA synthesis. So pyrosequencing is based on the detection of this pyrophosphate. Detection of this pyrophosphate using 4 different enzymes as a sequential enzyme in the pyrosequencing technology and the sequential addition of DNA nucleotide during this enzyme action. So it involves addition of various components during PCR reaction like DNA template for DNTPs, out of which one is the deoxyadenosine alpha thiosulfate, which is not utilized by one of the enzymes in the pyrosequencing reaction. 4 enzymes, that is DNA polymerase, ATP sulfurylase, luciferase, and O-pyrase. The enzyme DNA polymerase, it is used to add the DNTPs during the DNA polymerization, and due to its action, the pyrophosphate is get released whenever DNTPs get added within the DNA sequence. The second enzyme, that is O-pyrase, it is used to degrade unincorporated nucleotide, but its rate of action is slow compared to the DNA polymerase action, so that sufficient DNTPs that we have added during the PCR reaction should be available for DNA polymerase. Okay. Apart from that, we are using the third enzyme, that is ATP sulfurylase, which causes conversion of APS into ATP using the pyrophosphate released during DNA polymerization. So whenever DNTPs get added according to the complementary of DNA sequence found on template, the released pyrophosphate during this action is utilized by ATP sulfurylase to convert APS into ATP. The last enzyme here is the luciferase, which uses the ATP synthesized by ATP sulfurylase to convert the compound luciferin into oxyluciferin with the emission of visible light, which is proportional to the amount of ATP synthesized during ATP sulfurylase action. So like this, there are 4 different enzymes they are used in the pyrosequencing. This enzyme acts sequentially in the pyrosequencing reaction to sequence the DNA template. Okay. The one of the component that is added here is the primer to provide 3' OH group for the addition of new nucleotide by DNA polymerase. So these are the various components we require for the pyrosequencing method. So here we can see the detailed overview of the pyrosequencing diagrammatic representation. This is a template DNA used for the DNA sequencing, and it has attached the primer as a sequence to provide 3' OH group for the attachment of DNA polymerase for the addition of new nucleotide by DNA polymerase. And in that PCR reaction, where we have added all these components, DNA template for DNTPs, DNA template for enzyme, luciferin, APS, and primer. In this PCR reaction, we are adding the different nucleotides sequentially. Okay. So first nucleotide that we have added here is the deoxyadenosine triphosphate. As we have seen here, the nucleotide which found on the template is G nucleotide. So because this added ATP does not found the complementary here, so and because of that it is not get added here, and it will be, because of not incorporation, it will be get degraded due to the action of enzyme, that is O-pyrase. Okay. So if any nucleotide is not get added due to its not found complementary on the DNA template, then it will be get degraded by the action of O-pyrase enzyme into adenosine monophosphate. Okay. The next nucleotide we have added, that is sequentially, that is deoxycytidine triphosphate. As we have seen here, that there is a guanosine nucleotide present on the template DNA. So the added cytidine triphosphate, which finds the complementary of guanine, will be get added during the DNA polymerase action, and DNA polymerase will release the pyrophosphate from this cytidine triphosphate. The terminal 2 phosphate will be released from the cytidine triphosphate as a pyrophosphate group due to DNA polymerase action. and then deoxycytidine triphosphate will be get added with the release of pyrophosphate. This pyrophosphate is then utilized by the next enzyme in the pyrosequencing reaction, that is ATP sulfurylase. It will use this release pyrophosphate to convert APS, that is adenosine monophosphate, into ATP. Okay. So whenever the DNTPs get added within the DNA chain during the DNA polymerase action due to complementary, it will release the pyrophosphate. The released pyrophosphate is sequentially used by the next enzyme in the reaction, that is ATP sulfurylase for ATP. So synthesized ATP is directly proportional to the addition of nucleotide. Okay. So if any nucleotide is not get added, then it will be get ultimately degraded by a pyrase enzyme action. Okay. After that, the next enzyme in the pyrosequencing reaction, that is luciferase. Whenever ATP is get synthesized due to ATP sulfurylase action, this ATP is utilized by luciferase. Okay, here one point is noticed to be noted, that the luciferase it use only ATP synthesized by ATP sulfurylase, because whatever ATP we are adding here in the sequential addition, that is deoxyadenosine thiosulfate, alpha thiosulfate, which is not used by the luciferase. So luciferase use only ATP synthesized by ATP sulfurylase. So whenever ATP is synthesized by ATP sulfurylase, this luciferase will use this ATP to convert luciferin into oxyluciferin. Okay, so ATP synthesized by ATP sulfurylase is utilized by luciferase enzyme to convert luciferin into oxyluciferin with the simultaneous emission of visible light, and the amount of this visible light is directly proportional to the amount of ATP synthesized by ATP sulfurylase, which in turn is dependent on addition of nucleotide by DNA polymerase, because this ATP will be get synthesized due to release of pyrophosphate, which is utilized by ATP sulfurylase. Okay, so the amount of visible light that generated during this 4 enzymatic reaction will be detected by CCD camera, and the output will be fed into the data feeding computer, where the sequence of the template is determined by which nucleotide will get added. Okay, suppose here first nucleotide that is added is C, because here C nucleotide is added get added within the DNA synthesis. So the this complementary sequence that found on the DNA template is G nucleotide. So like this, we are determining the nucleotide sequence on the template during the pyrosequencing technology, which uses 4 different enzymes, that is DNA polymerase, ATP sulfurylase, luciferase, as the 3 enzymes that act sequentially, and a pyrase enzyme which degrade unincorporated nucleotide during this sequencing reaction. Okay, so DNA sequencing by pyrosequencing overall involves the steps like first preparation of PCR reaction mixture, which contain template DNA, 4 DNTPs, one of these deoxyadenosine alpha thiosulfate, primer sequence, 4 enzyme in the sequencing reaction, DNA polymerase, ATP sulfurylase, luciferase, a pyrase, a compound luciferin and adenosine monophosphate. Okay, then it involves the sequential addition of selected nucleotide, and involve the reaction of 4 different enzymes, that is the release of pyrophosphate if nucleotide will be get added due to the action of DNA polymerase. Okay, or it will be get degraded if it does not get incorporated into the DNA chain by the pyrase enzyme. Okay, so whenever it is not incorporated, it will be get degraded by a pyrase enzyme if added nucleotide is not incorporated in the DNA chain due to mismatch or non-complementary. Okay, after that, the conversion of APS into ATP by enzyme ATP sulfurylase using pyrophosphate release during the DNA synthesis. Then conversion of luciferin into the oxyluciferin by luciferase enzyme using ATP synthesized by ATP sulfurylase action with release of visible light proportional to the amount of ATP synthesized by ATP sulfurylase. Okay, so these are the basic steps involving the pyrosequencing. I hope you have understood the pyrosequencing very well. If you have any questions, you can comment me in the comment box. Thank you.